Sam Kean, The Violinist's Thumb, and Other Lost Tales of Love, War, and Genius, as written by our genetic code. Are you intrigued by the mysteries locked within our genetic code? Dive into the captivating world of DNA with Sam Kean's The Violinist's Thumb summary. This book takes you on a thrilling journey through the history of genetics, starting with the groundbreaking discoveries of Friedrich Miescher and Gregor Mendel. Explore how DNA shapes our physical traits, plays a role in evolution, and even impacts seemingly unrelated aspects such as artistic abilities, disease treatments, and genetic engineering. Uncover fascinating stories of scientific breakthroughs and open your mind to deeper insights into the building blocks of life. Unravel the marvels of our genetic heritage and the extraordinary potential that lies within our DNA.
The discovery of DNA and genetic inheritance. In the 1860s, Swiss biologist Friedrich Miescher discovered deoxyribonucleic acid, DNA, while studying white blood cells. However, nobody appreciated its significance at the time. At the same time, Gregor Mendel, a monk, was studying the heritability of traits in the common pea plant. Mendel discovered that traits do not blend together but rather some traits are dominant and others are recessive. He also found that traits are separate from each other, and they are controlled by discrete, separate factors known as genes. Unfortunately, the scientific community did not recognize the importance of these findings at the time. The emergence of DNA and the theory of evolution. Around the turn of the 20th century, Mendel's and Miescher's discoveries began to unveil their importance. Chromosomes were found to play a significant role in inheritance, and they were discovered to be made of DNA. Hugo de Vries found mutations in his primroses and theorized that species went through rare but intense mutation periods that produced new individuals with different traits, contrary to Darwin's theory of evolution. Thomas Hunt Morgan experimented with mutations in fruit flies and found that genes dictate the traits that creatures have, and mutations in those genes produce small changes in the creature's traits. These changes eventually lead to creatures that have beneficial mutations for survival and procreation, eventually spawning new species, in line with Darwin's theory of natural selection. The importance of DNA. DNA, discovered in the 1940s, plays a crucial role in making proteins that build cells, tissues, and organs. Only 1% of a DNA strand stores the recipe for making proteins, while the rest, once thought to be junk DNA, regulates genes and is crucial. Mutations in the blueprint can produce different traits with both beneficial and harmful effects on the organism's survival. Evolutionary breakthrough. Life on Earth likely began in the ocean near volcanic vents. The first life forms were single-celled simple organisms called microbes. They couldn't evolve beyond this stage as they lacked an efficient means of generating energy. However, evolution led some microbes to become large predators that could digest smaller ones, while others shrunk, became virus-like and began to get inside larger microbes and kill them. A breakthrough occurred when one large predatory microbe swallowed a small virus-like one, but it turned out neither could kill the other. The two began to coexist, creating more such partnerships as time went on. Eventually, the smaller microbes specialized in producing energy from oxygen while the larger ones provided raw materials and shelter. This combination was so efficient at generating energy that it enabled more intricate features to develop. The theory is that this evolutionary breakthrough can be seen in our cells, which contain mitochondria, the power plants of the cell. The miracle of multicellularity. The evolution from single-celled organisms to multicellular structures was a turning point in evolution. A sticky accident helped create specialized cells, paving the way for life as we know it. In humans, different cells develop from stem cells by muting unwanted genes. Hox genes, found in most animals, regulate the proper placement of specialized cells. Disrupting these genes leads to incomplete or deformed organisms. Our viral inheritance. Humans share genes with various organisms, including viruses. Some of the viral DNA present in our genome has become essential, such as the one responsible for the development of the placenta in mammals. Atavisms, an indication of our animal history, occur as a result of interrupted muting of genes during fetal development. Have you ever wondered how much of our genetic code is shared with other living organisms? The answer might surprise you. In early stages of their development, human, bird, fish, and lizard embryos share many similarities. This is because a lot of our genes are shared among various animals. However, as a human fetus develops, it begins to mute reptilian, avian, and other animal genes, while turning on mammal-specific genes, causing the similarities to fade. Yet, in some rare cases, the muting process is interrupted, resulting in an atavism, an evolutionary throwback whereby a trait that has disappeared long ago reappears. For example, some children are born with tails, a clear indication of our animal history. Interestingly, not only do animals share genes with us, viruses do too. In fact, approximately 8% of the human genome is of viral origin. Although most of this viral DNA is considered junk, some of it has become essential. One such example is the placenta, which connects the fetus to the mother in nearly all mammals. The development of the placenta has been a game-changer for reproduction, and the reason why placental mammals can be found all over the world. It's much easier to nurture future offspring when they are inside you, rather than in a vulnerable nest or on the ocean floor. Some researchers believe that we have viral DNA to thank for the development of the placenta, as the embryo latches onto the uterine wall using the same DNA that a virus uses when it latches onto a cell before injecting its own DNA. In a way, we have reappropriated this parasitic DNA for our own use. In summary, humans share genes with various organisms, including viruses, and some of the viral DNA present in our genome has become essential. Interrupted gene muting during fetal development can result in atavisms, providing a visible indication of our animal history. The mad scientist's Humanzee experiment. Explore the story of Ivanov, a Soviet scientist, who tried to create a human-chimpanzee hybrid in a bizarre experiment approved by communist officials. In the 1920s, Soviet scientist Ilya Ivanovich Ivanov embarked on a mission to crossbreed humans and chimpanzees. The experiment was conceived as a perfect way to insult religion, which communists perceived as their enemy. Ivanov tried inseminating a female chimp with human sperm and turned to inseminating a female human with chimp semen when his earlier attempts failed. However, he was detained by the Soviet secret police on obscure charges just before the experiment was conducted, and he died of a brain hemorrhage a day before his release from detention. Although there is evidence both for and against the possibility of human-chimpanzee hybrids, most scientists today agree that such a crossbreed would not be viable due to the vastly different sets of DNA in humans and chimpanzees. This bizarre episode in history raises interesting questions about the limits of scientific inquiry and the ethical implications of playing with nature. The Evolutionary Advantage of Art. The size of our brains allowed humans to become artistic, and the reason behind it might be due to evolutionary advantage. Humans stand out from other animals because of the size of our brains. But how did our brains get so big? Mutations played a crucial role in strengthening our neurons and expanding the cortex, the part of the brain involved in conscious thought. Less obvious mutations also contributed to our large brains, such as jaw mutations that made our skulls thinner, creating more space for the brain. One mutation caused us to lose a DNA segment that inhibited neuron growth, which led to bigger brains but also an increased risk of brain tumors. While we tend to think of artistic ability as a display of our brain's power, it might predate our species. Chimpanzees can be taught to paint and become passionately absorbed in it, even skipping feedings to pursue their art. One hypothesis for why humans became artistic is that it has a social role to strengthen community cohesion and overall survivability. Another theory is that art is a method of drawing attention to oneself, like a peacock's feathers, to attract mates. Singing and dancing showcase physical fitness, while poetry and painting display mental prowess. Artistic performances, therefore, became a sexy way to pass on artistic genes. The evolution of the human brain and its natural artistic inclination is an intriguing topic to explore. While artistic ability seems unique to us, it might have its roots in other animals and serve specific evolutionary purposes. Understanding DNA through the Human Genome Project. The completion of the Human Genome Project, HGP, in 2003 was a significant milestone in DNA sequencing aimed at pinpointing genetic causes of diseases to find appropriate medication. However, despite confirming that humans have only 22,000 genes compared to other species, and minimal genetic diversity, the project failed to deliver gene-based cures for diseases due to complexity and epigenetics. The environment's ability to switch genes on and off makes it difficult to understand the genetic causes of diseases. The completion of HGP brings us closer to the controversial realm of genetic engineering, splicing DNA among different species for desirable traits. As we approach these possibilities, the challenge arises concerning how we should handle these exciting yet potentially disconcerting prospects.
As we reach the end of our exploration into the world of DNA with Sam Kean's The Violinist's Thumb, we understand just how crucial genetics are in shaping our lives, both collectively and individually. From the minute microbes that marked the beginnings of life to the development of multicellular organisms, the book uncovers the intriguing connections between different species and the extraordinary phenomena of mutation and evolution. We've tackled significant scientific breakthroughs, gleaned insights into the human genome, and even navigated controversial discussions surrounding genetic engineering. Ultimately, The Violinist's Thumb offers a fascinating and accessible look into the incredible complexities of our genetic code and the endless potential it offers.